In this chapter, we'll explore the muscular system. The muscle functions to do two basic things. First of all, muscle tissue contracts. It gets shorter. As a result, movement is produced. The other thing is all muscle tissue generates heat. This is a byproduct of muscle activity. In order for muscle to contract, we have to break down ATP, and ATP breakdown yields some heat. There are three types of muscle: skeletal, smooth, and cardiac. Skeletal muscle is striated or striped. It's also voluntary. You consciously control when this muscle contracts. The cells are long and cylindrical. They run the entire length of whatever muscle they're in. They also have multiple nuclei, which has been pushed to the edge of the cell. Skeletal muscle is capable of producing powerful contractions. These muscles are attached to the skeleton, so when they contract, bones are going to move. These muscles also help us maintain our posture. While these muscles are very powerful, they do tire easily and they need to rest between contractions. Smooth muscle is non-striated muscle. There is no stripes or striations in this muscle, and it's involuntary. It's controlled by the unconscious part of your brain, the autonomic nervous system. We find smooth muscle in the walls of hollow organs, and when it contracts, it moves material through those hollow organs. Smooth muscle cells have a single nucleus and are spindle-shaped, so they're not very big cells. They also tend to arrange themselves in sheets. Around this tube, we have one sheet that's going around circularly and another sheet that's going longitudinally. Smooth muscle contracts very slowly, but contracts for a longer period of time. As a result, it tends not to tire very easily. Cardiac muscle has the same striations that we see in skeletal muscle, but this muscle is involuntary. You cannot control the contractions of your heart. Cardiac muscle is found only in the wall of the heart, and it's composed of these branching cells. These cells are not long and cylindrical, but branch, and they're held together by these junctions that are called intercalated discs. The heart muscle contracts at a steady rate, but is capable of increasing its contraction rate if necessary. When the heart muscle contracts, you're going to pump blood, so blood is the thing that is being moved by this muscle contraction. Cardiac muscle works long and does not tire easily. Most of our focus is going to be on the skeletal muscle because this is the one we control. As we look at the structure of a skeletal muscle, we're going to start calling things different names. A muscle cell is now going to be called a muscle fiber. So this muscle fiber is an individual cell in the muscle. Long, it runs the entire length of the muscle. It is surrounded by a connective tissue called the endomysium. Several muscle cells or muscle fibers are bundled together in a structure known as a fascicle, and the fascicle is surrounded by a connective tissue known as the perimysium. The entire muscle is covered by another dense fibrous connective tissue that is called the epimysium. So besides the muscle cells, there's an extensive amount of connective tissue in muscles to help support them and give them some additional strength. The epimysium is composed of regular dense connective tissue, and these fibers come and extend from the end of the muscle and help make up the attachment structures for the muscle to the bone. A cord-like structure, as you see here, is known as a tendon. If the muscle is flat, however, these fibers will come off in a sheet-like structure known as an aponeurosis. Both of them are responsible for holding the muscle to the bone, and they're both made of dense fibrous connective tissue. In order to understand how muscle cells work, we're going to have to look at the microscopic anatomy. Here is another name change. The plasmalemma is going to be called the sarcolemma. Myofibrils are the organelles that are in the muscle fibers that function in contraction. Each myofibril is filled with myofilaments. These myofilaments are cytoskeletal structures. The myofilaments within the myofibril overlap, and that's what makes the striations. Sometimes we have all of the myofilaments together in one spot, and that makes a darker looking line. And in other places, we only have one set of myofibrils, and that makes the lighter line. The myofibril is subdivided into structures known as sarcomeres. The sarcomeres are the contractile units of the muscle fiber. Because the myofibrils completely fill the cell, this is what pushes the nuclei to the side. The endoplasmic reticulum is now the sarcoplasmic reticulum. This structure stores calcium that's needed for muscle contraction. The sarcoplasmic reticulum is arranged in the cell in such a way that it wraps around the myofibrils. Here you see a skeletal muscle cell or fiber. This is the sarcolemma or the plasma membrane, and all of these are the myofibrils, all of these units, these organelles. Here you see the nucleus, they're pushed to the side. Now as we look inside a myofibril, here are those myofilaments. We have thick and thin filaments, and you can see sometimes the thick and the thin are overlapping together, and other times we just have places where we have the thin filaments, and that's what gives us our striations. A sarcomere runs from what we call Z-line to Z-line. These thin filaments are anchored here, and if we can shorten this somehow, we will pull these Z lines toward the M line, this middle line here, and that will shorten the sarcomere. The sarcomeres have two major types of myofilaments in them. The myosin is the thick filament made of the protein myosin. Myosin is what is going to need the ATP for proper contraction. On the myosin filament, there are these projections that are called myosin heads. The myosin heads are going to break down the ATP, and they're going to attach to the thin filament or the actin during muscle contraction. The actin is the thin filament made of the protein actin. Actin is controlled by two other proteins known as troponin and tropomyosin. So as we look at the arrangement of the fibers in a sarcomere, these thin actin fibers are the ones that are attached and anchored to the Z line. And these thicker myosin filaments have the little bumps on them, the little myosin heads. The actin overlaps wherever there are myosin heads. Here is another look at a muscle cell, several myofibrils. You can see the sarcomeres. And here is the arrangement of the sarcoplasmic reticulum. And there is a special feature to skeletal muscle. The sarcolemma, or the cell membrane, has these deep invaginations that are called T-tubules. So the cell membrane actually projects deeply into the cell, comes back out, and continues. And this is an important feature of how muscle cells function. In order for a muscle to contract, it has to be told to contract, and this is a function of the nervous system. One neuron, one nerve cell, and all of the individual muscle cells that nerve cell stimulates is known as a motor unit. Where the nerve communicates with the muscle cell is known as the neuromuscular junction. The nerve does not physically touch the muscle cell. There's a little space between the neuron and the muscle cell. The very end of the neuron is known as the axon terminal. This is the process that extends off the neuron. This is what forms the junction with the sarcolemma, the muscle cell membrane. The sarcolemma, just underneath the axon terminal, has receptors on it for the neurotransmitter acetylcholine. The space between the axon, the neuron, and the sarcolemma is called the synaptic cleft. So there is no direct touching of the axon to the cell membrane of the muscle cell. There's a little space, and this acetylcholine is released into the space, diffuses across, and attaches to the receptors on the muscle cell. Here is one nerve that has two neurons in it. This neuron sends axons to two muscle cells, so this is one motor unit. This neuron sends axons to three muscle cells, so that's another motor unit. One neuron will affect only the muscle cells in its motor unit. As the axon, that end of the neuron, comes down, it comes very, very close to the cell membrane of the muscle cell, but does not directly touch it. The axon will release the chemical acetylcholine, which will diffuse across this gap known as the synaptic cleft and attach to receptors on the sarcoplasm, the cell membrane of the muscle cell. When the nerve impulse releases that neurotransmitter, the acetylcholine, it's going to attach to receptors on the sarcolemma of the skeletal muscle. This makes the sarcolemma permeable to sodium. Now, sodium is normally pumped out of the cell and kept out of the cell. So if we make the membrane permeable to sodium, sodium is going to rush in. As sodium rushes into the muscle cell, it's going to generate an electrical condition called an action potential. This is going to travel over the entire sarcolemma. Remember, electricity is moving charged particles, and sodium ions are charged particles. Now, in addition to the sodium moving in, we're going to open up potassium channels, and potassium will move out of the muscle cell. As a result, we will stop that action potential and start returning the muscle cell back to its normal condition. The sodium and potassium pumps are what return the ion balance back to normal. So as we look at the steps, acetylcholine is released into the synaptic cleft. When it attaches to these receptors on the sarcolemma, it opens up these channels that allow sodium in and then potassium out. So sodium comes in first, that starts the action potential, then potassium sort of goes out and reverses the action potential. Those T-tubules, those impocketings of the cell membrane down into the cell, carry that action potential down into the cell. And these T-tubules are very close to that sarcoplasmic reticulum, so that when this area becomes excited, it irritates the sarcoplasmic reticulum membrane and it releases calcium into the cytoplasm of the cell. We're going to need calcium for the muscles to contract. So that action potential going down stimulates the sarcoplasmic reticulum. We release calcium. Calcium is responsible for myosin being able to bind to the actin molecules. Once we have the myosin binding to the actin, those myosin heads attaching to the actin, because the myosin heads have been cocked, they've been charged by the breakdown of ATP, they're going to make what's called a cross bridge. And then they're going to flex, and as they flex, they're going to pull the actin toward the center. This is going to shorten the sarcomere. Now, troponin and tropomyosin are what have to be arranged differently. Normally, this molecule of troponin is blocking these myosin binding sites. When calcium attaches to this protein, it opens up these myosin binding sites. This will allow myosin to attach to actin. That's called cross-bridge formation. And because this myosin head has been cocked, it's been charged, that was what we needed the ATP for, it's going to have that energy to power stroke. It's going to pull the actin toward the middle. Then if there is ATP present, it will recharge the myosin head, causing it to detach, go back into its cocked position, and as long as there are myosin binding sites on the actin, it will reattach and power stroke. So we're going to continue to move that actin toward the midline. That's going to shorten the sarcomere. That's going to shorten the muscle. Relaxation occurs when the action potential ends. There is no more acetylcholine delivered into that synaptic cleft, so we don't irritate the sarcolemma anymore, and so we don't irritate the endoplasmic reticulum or the sarcoplasmic reticulum. As a result, the calcium ions return back to the sarcoplasmic reticulum. This is going to cause the myosin binding sites to be blocked by troponin. We can't make any cross bridges, we can't do any flexing, so the muscle cell will return to its resting length. This is relaxation. Muscle cells respond to the all or none principle. They either contract fully or they don't contract at all. Now looking at an actual muscle fiber here, this is what's going on. This is in its relaxed state, no cross bridges, everything's in its regular position. And this is in its contracted state. We have had cross bridges formed, we have pulled the actin molecules to the middle, and notice we've lost some of those striations because we have more overlapping fibers. So there's the relaxed state, contracted, relaxed, contracted. This is why we call this the sliding filament theory, because the filaments slide over each other.